So 75 was the class, the overall average between the two classes. You guys were had an average that was 1/10 of a point higher than the second section. So and your median were the same. There were the second section had the 100. You guys had 396 and stuff. So it's the most even two sections I've ever taught. It's kind of astonishing. Okay, here comes some more. Who we have here? Lindy? So, I'm going to hand out the answer sheets, and we'll talk about the item analysis that I did, and then we'll move on with our stuff. Renee, I graded yours by hand, obviously. That was my hand graded. Grace? Katie? Anthony? Allie? Kiki? Whoever's name I call always seems to be on the other side of the room. We're okay. I need you. Blair. Ellie. Chase. Carly. Isabella. India. Connor. Michael. Andrew. Maria. Walden. Addison. Okay, that's all I can get back to that. All right, so let me go ahead and go over the item analysis. You could have gotten back as many as, I think it was 16 points. Maria. And some people did get back 16. There was only 2 people in the class who didn't get back any points. One of them made a solid A, the other one made the 100. They made 100 unadjusted by item analysis, so it was pretty darn impressive. I don't recall that ever happening on first exam before. As exams go, you guys your average is 2 points higher than last year's first exam, and 2 points lower than the year before. So you're right in the middle. So the results are a very flat bell curve, but a bell curve nonetheless. Obviously this was a doable test. There were, 21 A's and B's and only 13 that fell below the C mark. 18 that fell below the C mark, so slightly skewed to the upper part. Let me do the item analysis, and then we'll talk a little bit about the exam itself and what it all means in the real world. Okay, so item analysis. If you missed any of these questions, and I'll add that at the bottom of the sheet, if you're looking at your sheet like this, there are little lines, little hash marks by the questions you missed. Some of them will have a yellow mark on them, okay? And so these are the questions. If you missed any of these, they should have a little hash mark at the bottom. Yours should have an X across the correct answer, actually, in yours, where I did it. And it should be marked with yellow, and you should have gotten back 2 points for each of the ones that were item analyzed. So here are the ones. I'll read this list twice to make sure that I didn't make any mistakes. I almost always make a mistake, and here's my deal. If I owe you 2 points or something like that, if I missed a couple of points, let me know. I'd love to get you the 2 points. If I accidentally gave you 2 points that I shouldn't have, then that'll be our little secret, okay? I take that as a penalty on myself, basically. All right, so here they are. Number 6, 11, 12, 19, 20, 21, 30, and 46. Those were all missed by more than half the class, and I thought, okay, yeah, I'll give them back. Is that okay? Oh my gosh, 2 hands went up. Yeah.
You said 21.
I did. Did you miss it?
Yes, sir.
Yeah, you said 6, right?
Holy Toledo. God, I must have been in a daze.
Well,
All right, let me look at your answer sheet. You didn't miss 12.
oh,
Yeah.
I thought you were-
You didn't miss number 6. See these little hash marks?
Oh, okay.
Okay, I'm not exactly sure. You did miss 21. Let me help you out. I'll return that to this class. Okay, so just again, 6, 11, 19, 20, 21, 30, 46. Okay? As I said before, I try to make these exams pretty challenging. The raw average was a 65, and so the item analysis brought up the overall average to a 75. And then I do item analysis because I think, to me, it's not absolutely the perfect way of adjusting scores, but to me, it's the fairest way. I can't just add points to, anyway, there's a real good philosophy behind this. One of the weirdest courses I ever took in college was one called Testing and Assessment, and all sorts of studies about how to justify or adjust scores on exams, as opposed to adding bonus points or curving or anything like that. The one that apparently wins out is what I do, the item analysis process. It's considered the fairest to everyone. And I've been over a little bit backwards in it because above all, I want you to learn as much biology as you can. And second to that is I want to be as fair to you as I possibly can. So if you have any questions about it, I'll be happy to discuss it with you. Sometimes people don't feel like they got back as many points as the person next to them or something like that, and that might be the case, but who knows what happens on the next exam, right? So, if you made an A or a B on the exam, you probably have a pretty good idea what you need to be doing because you've been doing it, okay? Am I right? If you didn't make as good a grade as you like, I have a couple of things to say. Number one, this is the first exam. I suggest that you compare the exam score that you have with the quizzes that we've had so far and see how they match up percentile-wise. And if they match up pretty well, in other words, if you had a 60-something on your on the exam and you've been making 6s or 7s on the quizzes, then that should tell you something. You need to put more time into the books. Here's the deal. If you think you're an A student and you made a C on this exam, I won't let this one exam, particularly the first exam, kill anyone, all right? I can't just make it totally go away, but if you feel like you're, as I said, if you feel like you're an A student and you didn't make an A, show me you're an A student. You got me? okay? And lab helps a lot, quizzes can help way more than you imagine, on your score, so just buckle down and do it. If you didn't do as well as you would like, and you never made use of the tutor, system that we have, make use of the tutor system. Drop in Monday, Wednesday, 6:30 to 8:30. Any questions about it? If you want to come by my office during office hours and look at the exam, I don't give the exams back because I have a big test question bank of test questions that I sort through when I put together the exams. But you can stop by my office and look over and see what you missed and see why you missed it. And if you have any questions, then I can explain it to you. So, any questions in general? All right then, that's one down. Let's see here. All righty. So, changing gears. Not really, because we talked about metabolism and a little bit about the thermodynamics on the last class before the exam. Sorry about that. Takes a minute. So what we're going to be shifting to today is talking actually about metabolism, specifically respiration. We won't get all the way through respiration today, but we'll get up. My plan is to get at least through the Krebs cycle, the citric acid cycle, and if there's time, I may go on beyond that. But and then on Thursday we will do the rest of aerobic and anaerobic respiration, and we'll also do photosynthesis as a shorter section. I'll add that we won't have class on Tuesday of next week. Okay, I have to go on a trip up to Charlotte, North Carolina, and I won't be getting back until Tuesday afternoon sometime. So we'll lose a class, and just depending on how we're doing before the next exam, I may have to make it up with a voice over PowerPoint that'll be on Schoology that you'll do on your own time, if you follow what I mean. If you've done any of that, kind of like taking an online one lecture's worth. I don't know yet if that'll be the case, but if it is, then that's the way we'll solve the problem. I don't have any choice but to go on the thing to Charlotte. I'm in a national study of the aging process. So anyway, respiration and photosynthesis. I think I've showed you this before, and I mentioned the kind of the yin-yangishness to the whole thing. That's to photosynthesis and aerobic respiration. Anaerobic respiration is kind of a side game that some organisms play. A lot of bacteria do it, a lot of single-celled organisms. You even can do some anaerobic respiration. I don't know if you're aware of that, but you do have the ability to do a little bit of anaerobic respiration, i.e. without oxygen. When would that happen? Exercising, yeah. Any runners in here? Okay, so you know what I'm talking about. At the end of a race, how sometimes you're running a little bit low on oxygen. Back in the day, I was a swim coach, and I always told my swimmers if they could get out of the pool at the end of their race without help, then they didn't swim hard enough. That's not exactly true, but nevertheless. So I'll talk about the way that we do anaerobic respiration somewhat later on. Anyway, photosynthesis, we said, photosynthesis requires basically three things, okay? It requires water, it requires carbon dioxide, and it requires what? Where do you have your African violet part? Right up under your desk lamp, right? You have your African violet by now, right? Okay, sure. So that light, carbon dioxide, and water, those are the three major requirements for photosynthesis. Now your plant may need some other stuff, some fertilizer and stuff, but just for photosynthesis, that's what's required. In the process, it releases a couple of things. It releases as a waste product, it releases oxygen of all things, okay? And of course, it also produces sugars, glucose among them, but sugars in general. And of course, the reason the plant does that is to store food for its own use later on, because it also can carry out respiration. The respiration process requires a couple of things. It requires an energy source, sugar, glucose, and it requires water, basically, okay? And it requires oxygen, so three things it has to have. So at any rate, here the reactions of photosynthesis and respiration, aerobic respiration, are the exact opposite of each other. They require the same amount of glucose or produce the same amount of glucose. They require the same amount of carbon dioxide or produce the same amount of carbon dioxide, as we'll see when we go through these reactions. So here's kind of the little roadmap that I usually put on these presentations. All right, so you've all been around a campfire at some point, right? Something like that. And you toss wood in there, and the cellulose in the wood, of course, is a very high energy material because it's made of what? The cellulose in plants is made of long chains of glucose, exactly. And so when you burn the logs in a campfire, they go up fairly rapidly, releasing a large amount of heat. You wouldn't want to go sit on the campfire, it would hurt, okay? And also light, so it's releasing energy in a couple of different forms of expression. It's also producing carbon dioxide and water. Of course, the carbon dioxide just becomes part of the air, the gases in the air. And actually, so does the water, because as water is released in that process, what happens to it? It's immediately vaporized, so it becomes just part of the humidity in the atmosphere, if you will. Obviously, our cells can't do this because we would burn up in a hurry if our cells did this. We have to do it in a very much more stepwise fashion, as is indicated over here. The way that there's always an energy of activation in these types of processes. We talked about that on Tuesday of last week. So the way that if you're burning the logs, the way that you get over that energy of activation is how? You either have a match or something which adds fire already burning, and that heats up the wood, or if you want to cheat, you could throw some some charcoal starter on there or something, and then it'll go up. But basically, you artificially raise the heat, don't you, to get the whole process going. And once some of the cellulose, some of the glucose in the cellulose starts burning, starts combusting, then the heat of the fire is going to continue until you run out of fuel. We're good? The way we do it in biological systems is the way we overcome the activation energy. First of all, the activation energies, they're there, but they're much smaller. And for the most part, they're overcome either by body temperature, our steady body temperature, and or by enzymes, like we talked about on Tuesday last week. Okay? Catalysts, enzymes. And then we do it in a series of very small reactions. I've told you before that in the first part of respiration, glycolysis, which is carried out by both aerobic and anaerobic organisms, glycolysis, there are 10 reactions involved in taking a 6-carbon sugar, glucose, and breaking it down to 2 3-carbon sugars. It takes 10 reactions to do that. But in the process, some high energy molecules are stored, okay? And that's from the energy that's released in those reactions. We'll see in a few minutes. Okay. All right, so here's the just kind of the stylized makeup of a mitochondria. And obviously, what are these little folds called? Cristae, okay? And thylakoids here in the chloroplast. You'll notice that we have all these diagrammatic enzymes and things lined up along the inner lining here. Again, I talked something about the value of folds and surface area in biological systems. It gives places for attachment of important things. So here's the mitochondrion. It's got all these enzymes lined up like so. And here's the thylakoid membrane in the chloroplast. And you can see similarly very similar molecules lined up along that membrane. You'll also notice, and this is really important, and you'll see why as we go forward with this, in both cases, some of these enzymes are actually pumping hydrogen ions. What is a hydrogen ion? What? Yeah, so all it is a proton, okay? Yeah. So it's, exactly, Lauren said it's missing its electron, and that's exactly right. So all you have in the hydrogen ion is simply a proton, okay? And so these protons, indicated by H+, hydrogen ions, are getting pumped from the matrix of the mitochondrion, or the interior of the chloroplast. They're getting pumped into the intermembrane space between the inner and outer membrane of the mitochondrion, or the space inside the thylakoid. And that becomes incredibly important. That produces basically it produces a concentration gradient. Remember we talked about that in diffusion and so forth. And the way that we use that gradient is the way that we produce ATP, and the way that plants produce ATP and a couple of other types of molecules as well. Okay, so, and again, just emphasizing the fact that the particular placement of these enzymes in both of these cases side by side allow the very efficient processing of one reaction, pass on the product to the next reaction, to the next, to the next, to the next, because they're right there side by side. Okay? So, a lot of energies are released. Here's the overall reaction for burning one of those logs in your campfire, okay, at least as it regards the glucose. A glucose molecule requires 6 molecules of oxygen, and in the process, it's going to release 6 carbon dioxides. Where did those 6 carbons come from? Oh my goodness, there's 6 carbons in that glucose, aren't there? Okay, this is a balanced equation, by the way. If you count up the O's and the H's and so forth, it balances. And it releases 6 carbon dioxides and 6 waters and energy, which we store, of course, in the form mainly of ATP. There's a couple other possibilities, but basically ATP. Oops. Yeah. And this happens whether you're talking about aerobic U or anaerobic E. coli in your large intestine. Okay, this is the same reactions that take place exactly. So here we go. Aerobic respiration. So you can see there's basically 4 stages to it. There's 3 big stages and 1 little one. The first big stage is glycolysis. You'll notice that in the absence of oxygen, some types of cells can actually carry out the same 10 steps as we do. Okay, it's not required for glycolysis. In the case of aerobic respiration, the end of glycolysis, you produce 2 pyruvates. I'll show you pictures of those in a minute. And what happens, the next step is called pyruvate oxidation. It's just a 1-step process. So some people say there's basically 3 stages to aerobic respiration: glycolysis, citric acid cycle, and respiratory chain. But there is this other little 1-step process that takes place and is necessary. So pyruvate oxidation, and then the citric acid cycle, and then the respiratory chain, also known as the electron transport process. And at the end of that, the glucose has been completely oxidized, in other words, completely broken apart, giving up all of its electrons that are useful. And the waste products are water and carbon dioxide. Oh, what was it that plants needed? Water and carbon dioxide, right? And the amount of energy that's produced is about 36 ATP, but I'll come back to that here in just a second. In the case of glycolysis, since that's as far as the E. coli I was talking about can actually get, then it's going to be incompletely oxidized, and the waste product that's produced is going to be one or another type of organic compounds. It may be lactic acid. It may be, actually, if anyone ever here, I know you're not old enough to legally do so, but if you ever fermented, make some wine or beer or something like that? Okay, well, it's okay. You will when you take organic chemistry. Dr. Bordas has everybody making wine over here. But basically, the organic byproduct, waste product, is ethanol, ethyl alcohol, which is the alcohol that you may or may not consume. And the only amount of energy that comes out of this, though, is 2 ATPs. But if you're an anaerobic organism living in someone's hindgut or in the bottom of the mud of a swamp or someplace like that, you don't really have a very active lifestyle, so you don't really need a whole heck of a lot of energy. So 2 ATPs per glucose is more than enough for those organisms. Now, the 36, here's the deal. 36, and I'll show you how the 36 ATPs are calculated. Some of those ATPs have to be used in the whole process, getting stuff into the mitochondrion from the cytoplasm of the cell, mainly is the way. But actually, you end up having a net profit of about 32 to 33, somewhere in that range, okay, depending on what type of cell in the body and organism and so forth. So this is really the true benefit. But 32 ATPs is a lot of energy from a glucose molecule. Yeah.
So what happens to other 4? They just get wasted?
No, like I say, they're involved in getting stuff across. So this process, glycolysis, I'm going to show you a slide in a minute. Glycolysis starts in the cytoplasm of the cell, okay? But Krebs cycle takes place inside the mitochondria and electron transport as well. And so just moving some of the intermediate products from one place to another accounts for most of the use of those ATPs. It's not a completely energy-free process. It doesn't cost a whole lot, as you can see, but it's still, there are 2 or 3 or 4 ATPs that get used up in the process of this. There's another stage as well. There's another thing. One of the stages of the citric acid cycle, I'll point it out when we get to it, doesn't initially produce ATP. It produces
this is GTP, guanosine triphosphate, as opposed to adenosine triphosphate. And for the most part, that GTP does get converted to ATP, but some of it gets used directly by some other cellular processes as well. So you lose some of the ATPs in those processes. So there's probably a couple other ways that those 4 ATPs disappear, but they're not wasted. They're put to use in one fashion or another. They are 36 are made in this process, but some of them do get spent. All right, so when we oxidize the material, the one we're starting with here is glucose, but each step of the process, you end up with a product that has to be oxidized, okay? It loses electrons. We've defined that before. Anytime in a chemical reaction, a product, a reactant gives up electrons, then we say that it's oxidized. The reactant or the product that receives those electrons is reduced, exactly, because electrons are negatively charged, so the charge of the molecule goes down. Usually the energy level will go up, as we'll talk about that. I want to point this out. Those hydrogen atoms, those hydrogen ions, the protons, okay? Usually when these electrons are moving from one molecule to another, there's usually in some fashion some hydrogen ions or hydrogen atoms or protons that are following them along. When I was a freshman, that just drove me crazy. And I'm making this point because I don't think my professor made that point. And I was just going, where the heck? And so if you get confused, don't worry. All of your professors were confused at one point or another as well. And I like to point out the times that I stumbled, and that was one of the times that I stumbled, was figuring out exactly what those protons were all about. So don't forget, anytime electrons are moving, there's going to be some hydrogen atoms possibly attached to a molecule or possibly just floating around in space near the process. So the electrons typically going to be transferred to something else. That something else will be then reduced, okay, as it says here. I'm trying not to read it, but pretty straightforward. And a lot of energy passes in this process, and we're going to see each of the steps. I'm not going to make you memorize each of the steps. I do want you to know what happens in each of the stages. So glycolysis, pyruvate oxidation, Krebs cycle. But later on, when you take biochem and or when you take cell biology, if you take those, you'll have to know the whole process, each stage, and probably have to draw the intermediate molecules as well. But that's for later. It's a thrill that you have yet to enjoy. But it's coming, don't worry. When I took biochemistry at University of New Hampshire, it was a graduate level, a PhD level biochemistry course. But we had to know so many pathways, it was just mind-boggling. And in those days, nowadays, printer paper is just like typing paper, mostly. But back in those days, usually no one had their own individual printer. There'd be 2 or 3 in the department, and they'd be these standalone devices with a paper that would come out. And the paper was this long folded bits of paper in these big boxes. And you know what I'm talking about? Is it making any sense at all? At any rate, it was like miles of paper, like serrated or perforated so you could tear the pieces apart. And so it would just print out. So it wasn't on a screen, whatever results, it'd be printing out the stuff that you were doing, your results. And so clever biochemistry students, who shall remain unnamed, might have occasionally gone by one of the computer centers and stolen about that much of that paper. And then whenever we were memorizing our reactions, that we had to know the structures and the names of the molecules and the enzymes and all that kind of stuff, we just sit there, write them, flip over to the next one, write them, flip over to the next one, write them, flip over to the next one. And so you get these big books of your study and stuff. So anyway, you'll get to that point sometime. It's kind of fun and kind of pain in the butt. So here we go. Here's a reduced compound. A reduced compound typically contains a substantial amount of energy. Glucose would be a very reduced compound, okay? When that compound is oxidized, it's going to give up some energy. So here's the oxidized compound A, whatever it may be. And here's the oxidized B, low energy. It's going to pick up those electrons, thereby being reduced, and the energy that goes along with them, okay? Some of the energy at least. There are a couple of what we call coenzymes, several of them in this process. One of the big ones is one called NAD, which stands for nicotinic acid dinucleotide. You can see where the dinucleotide comes from if you remember what the shape of a nucleotide is. See these 2 things? It has an oxidized form, NAD+, and a reduced form. So reduced would be the one with the extra energy, right? So here's the reduced form, and you can see they're very, very similar molecules. This one, however, contains enough energy to make about 3 ATP molecules, okay? We don't use the NADH, it's really too high energy a molecule for us to use in most of our cellular processes, okay? 3 ATPs at once is a lot of energy. So we use it to make ATPs, and then we use the ATP because we want to wiggle a muscle or blink our eye or whatever process requires energy. So that's one of the big ones. There's a couple others. There's one called FADH2, and ATP is actually an activated carrier molecule or electron carrier as well. So again, here's the NAD. Here's the highly reduced compound, and you hear those protons I was talking about, the hydrogens, right? And it's going to give up those hydrogens. In this case, the NAD, the oxidized version, is going to get reduced to NADH. And so there's one of the H's. And here's the other. Remember I said sometimes the other proton is just sort of sticking around in the vicinity, all right? And so there it is. And again, you can see the process. You'll see, I'll show you charts like this fairly frequently. I already have. So here are the pathways that are involved in aerobic respiration. First of all, there's glycolysis. I'll show you these in more detail in a minute, but at glycolysis, we start with the glucose. It actually, the process of this is, it doesn't show up in this chart, but the process of this is to produce ATPs, right? That's what's usable, right? It actually takes a couple of ATPs and some ADPs to get this going, all right? The ATPs basically do the same thing when you lit your campfire with a match, so to speak. The ATPs pretty much do the same thing in the glycolysis pathway. You use 2 of them to get the process started. And then you have to have NAD+, and in the process, there's 10 reactions. You're going to start with 1 6-carbon glucose. You're going to end up with 2 3-carbon pyruvates, aka pyruvic acid, okay? And you're going to produce a couple of ATPs, a net gain of 2 ATPs. You're going to produce a net gain of 2 NADHs, again, high energy molecules, and you're going to produce 2, as I said, 2 pyruvates. And I'm not going to go through all the stages here. This is a really good thing. I've told you a couple of times that you ought to, only half jokingly, you ought to, if you get a color copy of something like this, and print it out and laminate it and put it in your backpack or whatever, and when you're sitting around with no social life because you're a science major, then you can pull it out and review it. This is the kind of stuff you need to know about. Okay, you need to be tight with this. Put it in your toolbox. So anyway, pyruvate oxidation produces another couple of NADHs, and then the citric acid cycle produces a total of 6 NADHs, as we're going to see, and then all those NADHs and the FADH2s enter the respiratory of the electron transport system, and we probably won't get to that today, but we may. So, any questions about the basic overall picture of aerobic respiration? We're going to get to it. Yeah.
How do you know which one produces, or how much ATP each one produces?
Each one.
Each
NADH. It's back in the 50s. There's a reason that the citric acid cycle is called the Krebs cycle. There was a guy. He doesn't really have this Krebs cycle named after him. He has a title appended to his name, Sir Julian Krebs. Okay, and he was the one that figured all that out basically in a cell biology / biochem lab with fancy equipment that basically could test exactly what each of these enzymes did and how much energy and so forth. So I would answer that in more detail, but I wasn't involved. I was only like 6 years old when that was going on, so that was a while back. But anyway, yeah, I tend to use the term Krebs cycle because Julian Krebs was a freakishly big deal in biochemistry for working all that stuff out. So, and this basically talks about what I was already going over. So there's NADH being produced by pyruvate oxidation, and we'll see these in some more detail in a minute. You have electrons, hydrogen atoms, electrons, hydrogen atoms, electrons, electrons, hydrogen atoms. They just go together. They're a team. Okay, so don't lose track of that. All right, so here's the pathway for 1 or 2 of the types of fermentation or anaerobic respiration. Once again, they go through exactly the same stage. You start out with the glucose. We go through glycolysis. Pyruvate's produced at the end of this thing. Also some NADH is produced. Also some ATP is produced. The pyruvate's the interesting thing, though, because an anaerobic organism can't use the pyruvate directly. So what does it do? It also can't really use the NADH directly. The only way we use the NADH is in our electron transport. We'll see this probably on Thursday. And to do the electron transport, there's one molecule that's absolutely necessary. Anyone know? Aerobic respiration? Oxygen, yeah. And anaerobic organisms don't make use of the oxygen, so they can't do the electron transport, all right? And our cells use the NADH to produce ATPs and electron transport, but an anaerobic organism cannot do that, all right? So, what happens here, and this is really important, anaerobic organisms have to ferment that pyruvate. The two types that we're familiar with, that you're familiar with now, are alcoholic fermentation, like I was talking about in wine and beer making. If you haven't made wine and beer, you've probably been around baking bread, right? With yeast, that's an alcoholic fermentation as well. When what happens in the process is alcohol and CO2, and who's ever made bread with yeast and all? What happens? It rises, doesn't it? Why does it rise? The CO2 is produced. Okay. If you smell the dough before you cook it, you may be able to detect the alcohol. It is also producing alcohol, but of course, when you bake the bread, the alcohol evaporates away fairly quickly. It doesn't produce that much anyway, but it is detectable. But the CO2 is what causes your bread to rise. And the other one is lactate, and this is what happens when. Earthly, I saw you raise your hand. I know you're a runner. This is what happens when Earthly's at the end of the race and she's winning and she's just got to stay in first place, right? Okay, and her muscles start hurting and she's starting to go like that. Okay, and she may be so fit that doesn't happen. But at any rate, what's happening there is that a lot of her cells, her muscle cells, are beginning to operate at least partially on an anaerobic process. And what they're doing is they're converting that pyruvate to lactic acid, but in the process they're taking the NADH and converting it to NAD. Now why is that valuable? What's required for glycolysis? NAD. And every time you go through glycolysis, what are you doing? You're using up your NAD supply, aren't you? Is everybody with me? Okay? If you run out of NAD, what can't happen? Glycolysis. You can't make those ATPs anymore. And so anaerobic respiration is all about regenerating the yeast that makes the alcohol and makes your bread rise doesn't care about that. What it's trying to do is get the NAD back, okay, so it can continue this process. So, and the lactate is what the athlete's doing to try to get some NAD back to continue the process. Now, for those of you that take microbiology, well, at least the microbial biology, I don't know how much Dr. Fox gets into this part, probably a good bit actually. There are some other forms, some other products that are produced in other anaerobic processes, and if you're taking microbial next semester with me, I'll get into that in more detail. In fact, we'll test for some of them in our lab exercises. At any rate, that's anaerobic respiration. So, a little bit of a comparison between, in general, between prokaryotes and eukaryotes. What did I tell you that the mitochondrion, in fact, actually is? It's an endosymbiotic bacteria, okay? And again, Dr. Lynn Margulis, the lady that figured out about endosymbiosis theory, okay, she worked that up, said, okay, if the mitochondrion is a bacterium, it should act like a bacterium in a variety of ways. And I already told you that what you find. Bacteria have a single circular chromosome. Mitochondria have a single circular chromosome. Bacteria have a prokaryotic version of ribosomes. Mitochondria have a prokaryotic version of ribosomes, okay? Here's another one, check it out. So, in eukaryotes, your cells, what happens in the cytoplasm of your cell is the glycolysis process. And if any fermentation needs to take place, it also takes place in the cytoplasm, not in the mitochondria. Inside the mitochondria, attached to the inner membrane, is where pyruvate oxidation takes place and the electron transport process takes place. The citric acid cycle actually takes place inside the matrix of the cell, which of course, when you consider that this is a bacterium, the matrix of the mitochondrion is what? It's the cytoplasm of the bacterium, isn't it? Okay? So in prokaryotes, what do we see? In the cytoplasm, we've got glycolysis. Fermentation takes place there. The citric acid cycle takes place. I already said that if mitochondria are bacteria, the matrix represents their cytoplasm. And then attached to the inner surface of the plasma membrane, the cell membrane, are the same things that are attached to that inner membrane in the mitochondria. Pyruvate oxidation and respiratory chain. So again, this is some really powerful evidence for the functionality of Dr. Margulis' endosymbiosis theory. You can kind of see why mitochondria are really alien organisms living in all your cells, but that's okay. So, all right, so glycolysis, here we go. We got to get over this energy barrier here. So we have a couple of ATPs that get this whole process started. We spin 2 ATPs here, okay? Starting with glucose at a lower energy level. It raises, the energy up to, I'm gonna name some of these, but I'm not gonna require you to know the names at this point. Something called glyceraldehyde 3-phosphate, okay? And then, glyceraldehyde 3-phosphate gets broken down, gets, oxidized, and in the process, it uses up 2 NAD pluses and produces 2 NADHs. And in the next step, a couple of ATPs get produced. That balances the budget for ATP, doesn't it? We use 2 ATPs to get started. We're producing 2 ATPs here. And in the last step here, there's 2 more ATPs produced. So the net gain of glycolysis in an aerobic organism is what? 2 ATPs and 2 NADHs. Don't forget those NADHs. They're important. Okay, they're, as I said, about 3 times higher energy than the ATP. So just for glycolysis, there's the equivalent. How much did I say NADH is worth? About 3 ATPs. So we got 6 ATP equivalents here, and another 2 that profit ATP. So we produced enough. We stored enough energy just with glycolysis to theoretically produce about 8 ATPs, right? We'll keep up with that calculation. So here are the stages. Again, you're not going to need to know these specifically here. You do need to know that it starts with glucose and that it ends with pyruvate, pyruvic acid. But all these stages along the way, they're just things that you'll be entrusted with later. So there's an ATP that gets the thing started. There's one that gets it going. And whoa, that was exciting. Put that nonsense out. There we go. So at any rate, as I said, you get glyceraldehyde 3-phosphate here produced, and then when it gets broken down into a couple of 3-carbon sugars, okay, these are 1,3-bisphosphoglycerate. Again, you'll know that later. But in the process, we get a couple of ATPs produced, and we get a couple of NADHs produced. And we go on down. There's a couple of waters produced here, and this is called metabolic water. We'll talk about that later on, especially when we get into electron transport system. And then in the last step, we produce a couple more ATPs, and we produce something called pyruvate. It's a 3-carbon sugar. You can see the carbon, maybe 1, 2, 3, attached to oxygen and hydrogen as sugars off and on. It would be a triose. Remember I described the distinction between trioses and hexoses and so forth. It's a high energy molecule. It's very high energy because at this point we've captured enough energy to produce 8 ATPs. The whole process, though, is going to release 36. 32-ish would be useful, but if you with it. Okay? So that's glycolysis. I do want you to know what goes into each of these stages and what comes out. So what went into glycolysis? Some NAD pluses, 2 NAD pluses, as a matter of fact, 2 ATPs, get started, 4 ADPs, and then what comes out of glycolysis, and 1 glucose. And what comes out of glycolysis? 2 NADHs, 4 ATPs, but you already spent 2, so a net gain of 2, and 2 NADHs, and some water. Okay, this is the kind of stuff that I want you to be conversant with, not the names of the molecules. Yeah.
You said like, you said 4 ATP come in and 4 ATP come
No,
out.
2 ATPs go in.
2 ATPs come in and.
Yeah, and 4 come out. So a net gain of 2. All right, pyruvate oxidation. Here we have this high energy pyruvate molecule. 2 things are required. Once again, an NAD plus is required, and it's going to be reduced to NADH. There's that proton tagging along that I keep talking about. And in addition, you're going to add it's called a coenzyme. A coenzyme is just what the name implies. What does the name imply? It's something that works with enzymes. Okay, it's an assistant enzyme, if you will. So this is coenzyme A, and it's going to play a role here. And in the process, a carbon dioxide is going to be released. So we started with a 3-carbon pyruvate. We end up with a 2-carbon, what do you call it, sugar at this point. It's called acetyl-CoA. And we've lost one of the CO2s. But for each glucose molecule, how many times did pyruvate oxidation happen? Don't sit there like you don't know the answer. For each glucose that started this, how many pyruvate oxidations happen? 2, exactly. So there's not just 1 NADH produced for each glucose, right? There's not just 1 CO2, there's 2 of each of those produced. And there's 2 acetyl-CoAs. You with me?
So from here on, everything, all the numbers are doubled, basically. So, the citric acid cycle, AKA the Sir Julian Krebs citric acid cycle, also called the tricarboxylic acid cycle. There's all kinds of names, but citric acid cycle or Krebs cycle are the ones that I tend to use. So these produce, as we'll see, some CO, the rest of the CO2s, some FADH2s, 3 NADHs, and 1 ATP for every acetyl-CoA. But how many acetyl-CoAs enter that cycle for each glucose? 2. Okay, so you double the numbers. And here it is. Once again, I would suggest that you might put this in your toolbox, or laminate it and put it in your backpack, or whatever. But this is the knowledge that you need to have, basically, for understanding the citric acid cycle. So what's required? A coenzyme A, some NAD pluses, some FADs, some ADP, because you're going to produce some ATP. And you're going to also, what's going to be released is coenzyme A. The coenzyme A, like other enzyme-related molecules, just gets used over and over and over. It doesn't get destroyed. You're going to produce 2 more CO2s, but stop and think about it. In the process of pyruvate oxidation, we released 2 of the 6 carbons that were on the glucose, right? This pyruvate oxidation happened twice, so that's 2 of the 6. We have 4 carbons left that enter the citric acid cycle. At the end of the citric acid cycle, each of those pyruvates has been broken down to 2 CO2s, but there were 2 pyruvates, all right? So how many carbons have we gotten rid of? We've gotten rid of all 6 carbons that were in that glucose molecule at that point. And here's the cycle. Again, you don't have to know it precisely, but remember, 1 pyruvate enters, acetyl-CoA enters, and it's called the citric acid cycle because the first thing it gets converted to is citric acid, hence citric acid cycle. And it goes through some others, isocitrate, alpha-ketoglutarate, and on and on and on. But in the process here, we release, we produce 1 NADH molecule, and we release a CO2. But since this cycle has to run twice for each glucose, this actually represents 2 NADHs, right? And 2 CO2s. The next stage, alpha-ketoglutarate going to succinyl-CoA, we also produce 2 NADHs and 2 CO2s, and that's the end of our CO2 supply. There were 6 carbons that went in. We lost 2 of them in pyruvate oxidation. We lost 2 of them here. We lost 2 of them here. That glucose is gone. All the energy at this point is either stored in NADH or ATP or some of these other molecules that are in the citric acid cycle. Next stage, succinyl-CoA goes to succinic acid. And as I said, there's a stage here in the citric acid cycle where it's not producing ATP directly, it produces GTP. It's an equivalent molecule. For the most part, our cells don't use it for energy needs, but they do for some things. So that's one of the places that some of the ATPs kind of disappear, if you will. But most of the GTP just simply gets converted directly to ATP. But how many ATPs? Come on, keep asking this. You know the answer. 2. There we go. A little class participation. That's what I love. All right. And so you get 2 ATPs. And then the next stage, from succinic acid to fumaric acid, this is this other weird activated carrier molecule. FAD gets converted to FADH2. But again, there are 2 of them. And FADH2 stores enough energy to produce about 2 ATPs. So it's not quite as high energy as NADH, but it still is a high energy molecule. And then we go through the rest of the stage. The last stage of the citric acid cycle here, malic acid to oxalic acid, produces another couple of NADHs because 2 cycles. And so let's add up our NADHs. How many NADHs did we get? What's our balance sheet right now? We had how many NADHs producing glycolysis? 2. How many were produced in pyruvate oxidation? 2 more because it had to run twice. So we've got 4 NADHs. Here we've got 1, 2, 3, but since it has to run twice, that's 6. So we've got 10 NADHs. That's worth how many ATPs? 30 ATPs right there, okay? And we got 2 more ATPs here, right? So that's 32. How many ATPs did we get out of glycolysis? 2, right? And so that's up to 34. And then the FADH2s produce up to about 38 ATPs. So that's the balance sheet. Again, some of those ATPs get used for other things
that kind of drive the process along. So here we go. The whole process all the way through the citric acid cycle. So glucose, 2 ATPs get used, 2 ATPs, 4 ATPs get produced, 2 NADHs get produced, pyruvate, 2 NADHs get produced. Citric acid cycle 1, 2, 3, 4, 5, 6 get produced, 2 FADH2s, 2 ATPs, and what happens next? All those NADHs and the FADH2s do what? Where do they go? They're going to go to the inner membrane of the mitochondrion and get passed along the electron transport chain, the respiratory chain. So we will get here today, good. All right, so here's the thing. These are the high energy molecules that are critically important in electron transport. They came from glycolysis, pyruvate oxidation, and citric acid cycle. What is going on here? Jeez. All right, this clip is not playing together. Let's see if I can handle it from over here, I guess. I don't know like this. All right, so what happens from here on? Some of it, most of the rest of what happens actually is attached to the inner mitochondrial membrane. Where did I say the citric acid cycle occurs? The Krebs cycle? It's in the matrix of the mitochondria, and it's not associated with the membrane particularly, but most of the rest of the things are attached to that inner mitochondrial membrane. And again, this is the diagram of the mitochondria with the crystine, the inner membrane, and the outer membrane. And here we go. Here's the electron transport. So here's what happens with a NADH. Okay, it enters the system. There's that proton tagging along. Okay, still, it enters the system. It meets up with the first of the so-called cytochromes. This one's an enzyme. Basically, it's a complex of several enzymes, but cytochromes. But basically, we call it NADHQ reductase complex. You don't have to know that. Just understand that a lot of these molecules that are part of the electron transport system are called cytochromes. So if I were to say something about a cytochrome on the test, I would expect you to know that that's associated with the membrane, the inner membrane, okay? But for the most part, that's not stuff that I'm going to require you to be on top of. So at any rate, so what happens here is that the NADH gets reduced to NAD, so it's basically reducing the NADHQ, isn't it? Okay, it's handing in electrons. If those electrons are going to be passed on to the next thing, ubiquinone. They're going to be passed on to the next reductase, cytochrome C reductase. They're going to pass along to cytochrome C, and at the end of that process, those electrons are going to be used, they're going to be combined with oxygen, and those protons I keep talking about. If you take a little bit of energy, electrons, and you take hydrogen, protons, which have been floating around, and you take the oxygen that you're breathing, oxygen, right? What do you have? Oxygen and hydrogen, water. Right? Can you see that there? Okay, and that's the end of the process. That's the overall system. How many of you have ever been through this before in high school or something a little bit? Okay, it's not that usual to get into much detail in this in high school, and so if you're feeling a little bit lost, look around. There were like 3 or 4 hands that went up at that point. A lot of other hands that didn't go up. So if you're feeling a little bit concerned about this, lost about this, don't be. You're where you should be. It is complex, and but you're going to have to understand the overall process. Again, I don't require you to see all the trees, all the individual molecules and enzymes and things, but I do want you to understand how the forest works. Kind of falling on that old analogy of not seeing the forest for the trees. I want you to see the forest. You'll worry about the trees later on. Did that make a little sense? Okay. Anyway, so at any rate, that's kind of the forest version of this. The trees are a little bit more complex. Each of those things, the NADHQ reductase, cytochrome C, and so forth, are what we refer to as proton pumps. So what do they do with that energy that they're getting from these electrons? Well, go. This is making me upset. Guess I'm just going to get stuck back here behind it. So they are proton pumps, and you'll see they're pumping those hydrogens into that inner membrane space. And as they do so, what's going to happen? You're going to build up a concentration of protons in that space. That creates two things. Number one, it creates a proton gradient, as I said, okay? And what else does it do? What do you remember about protons? We talked about protons in a completely unrelated, not completely unrelated, but somewhat unrelated matter when I was talking about chemistry. What happens when you build up protons in a material, in a solution? It becomes more acidic. There we go, okay? And so if that continued without any kind of relief, they just flash. Maybe something wrong with the system. At any rate, what happens is, if they just keep going in there without being released in some fashion, then you run into a pH problem in the intermembrane space, wouldn't you? Enough possibly to damage or destroy the mitochondria. So what happens is that we use those, excuse me, we use that electron gradient. It's actually referred to as the proton motive force. If you've never heard that term, but it does produce a force. Yeah, well, there it is, proton motive force. And one of the best molecules in the world, I've told you about one other biological structure that's rotary, that just spins. What is that? The bacterial, the prokaryotic flagellum. There is a biological, a biomolecular structure, an enzyme, ATP synthase, which apparently also spins, okay, in a rotary fashion. And so sitting there on that inner membrane is this enzyme. It's also a channel protein. And as those protons build up in the inner space, the intermembrane space, they pass down through it's almost like a carousel or a turbine at the bottom of a dam, okay? And as it happens, the energy from that proton matrix gradient, excuse me, is actually the energy from that takes ATPs and inorganic phosphates and converts them to ATPs. Okay, that's the way that the process happens. And again, to me, so far I haven't even really mentioned oxygen, except when I was showing you that overview. The oxygen molecules you're breathing in right now don't get used until the absolute very dead last stage of aerobic respiration. And they're used to combine with the protons that have been, as the protons move into the matrix, we still got the problem of potential buildup of acidity, right? Okay? So still got to do something with those protons, and what we do with them is we combine them with some of the electrons and with the oxygen that you're breathing to make water molecules. And what happens to water molecules? They basically become part of your water budget. I mean, you have to have a certain amount of water every day. And so the extent that you're not dehydrated, it's just going to end up being part of your metabolic water that you use. There are a lot of insects that live in the desert and never ever see a drop of water in their entire lives. They are able to get by on what we call metabolic water. When they carry out aerobic respiration, they produce a substantial amount of water molecules, and that's what they live on. Any of you who've ever been around any kind of racetracks like horses or dogs or something like that, you may or may not be aware that at the end of a race, a really hard race, there's something called urination reflex. Are you familiar with this? They pee. Why? Because they just used up massive amounts of glucose stored, and at the end of the process, they obviously took a lot of energy, oxygen to run the race, and at the end of that, they produced a lot of metabolic water, and so something has to be done with it. Funny story. My oldest son's girlfriend, Nikki, back in the day, I like Nikki a lot. I'm very happy with his current wife, but she was a girlfriend back in high school days, basically. Nikki had one of the best jobs ever, okay? She worked at the dog track. I don't know if we still have a dog track here in Mobile, but we used to. And she had a job there, and her job was after each of the dogs finished racing, she and some other, basically older high school age kids, would go with little pans and get those pans under those dogs, okay, to catch their urine. What in the world was that about? Why do they want to catch it? They just don't want to pee it on the track? What? Boom. Yeah, they were to check for the urine was then sent to the path lab in South Alabama, and it was tested for various types of performance-enhancing metabolic products, okay? And you'd be surprised how often they came up with performance-enhancing metabolic products. And she got paid. This is like a couple of decades ago now. Yeah. She was getting paid like $20 an hour or so for a high school job. This was some sweet bank, all right? But she would go over to our house afterwards, and she just basically crash are gonna collapse, melt under our couch in his exhaustion, because, well, she just been chasing down gray hounds all evening and trying to hold them still and get a tray under them so they could pee in it. Same thing happens at a dog track. The same thing happens with humans as well. If you watch the Olympics, things like steeplechase and all, they don't televise them that much because they're kind of hard to follow. And honestly, after you watch someone run 5 miles, it's like, whew, let's check some checkpoints along the way. But in the Olympics, of course, the runners are doing the same thing, aren't they? And are they stopping at porta potties along the way? What are they doing? How are they doing with all that metabolic water? They just pee as they run. Yeah, exactly. And so one of the fun things when I watch the Olympics, I love the Olympics, okay? I can't wait until next summer. My big sport's swimming, but I love a lot of the other sports. And I love the long races because if you watch real closely, when they show pictures, sometimes you'll see the runners will have like this glistening on their legs or something. As a biologist, I appreciate what they've done for their hopefully gold or silver or bronze medal, right? Pee on the way. No one ever says anything about that, but nevertheless, that's what happened. So, there it is, oxygen and water. Let's see here. All right, so here we go. Here's, if you could get really, really tiny, shrink yourself down to a microscopic size, and get inside a mitochondria, and here's what you'd see. There's the NADHQ reductase, taking NADH, producing NAD, pumping the protons out in the intermembrane space, ubiquinone, cytochrome reductase, so forth and so on. And then here's this fabulous ATP synthase. I think ATP synthase, Science Magazine basically names a molecule of the year each year. It's a big science publication. And I think the second molecule, or the third that they named as molecule of the year was ATP synthase. You're going to see ATP synthase again when we look at photosynthesis, okay? And while it may not be absolutely the same molecule, it is similarly structured and similarly rotary type structure. So anyway, at the end of this, the protons, the proton motive force flow back down through here. The energy of that, just like the water passing through the turbines in a dam, the energy of those protons passing through this turbine here the energy of that's used to produce more ATPs. And that's the end of it, except for the production of the water at the end of it. So fermentation, I've gotten farther today than I anticipated, but we're going to go ahead. This probably means that you might get a little bit of a break on Thursday, or I might move on to the next topics if we're going to miss a day next week, but we'll try that on the sides when we get there. So let's sum up fermentation. This is anaerobic respiration, and once again, the glycolysis is exactly what happens in your glycolysis. Okay, same reaction, same enzymes, the whole work. And it produces 2 ATPs net gain, and it produces 2 pyruvates net gain. And if you're anaerobic and you can't access the electron transport where you use those NADHs, you got to do something with them to get the NADHs back. And so what happens is it really depends on the organism. If you're talking about yeast or some of the bacteria that carry out alcoholic fermentation, then what they do is they use the energy from these NADHs. There's a couple of other reactions that take place here, but they use the energy from those NADHs to convert this intermediate into ethanol. That's the alcohol that's in beer and wine, and then also the carbon dioxide here is what makes your bread rise as you see it. That's also what makes if you have champagne and beer that's bubbly, that carbon dioxide is at least traditionally is what makes beer fizzy, champagne fizzy, and so forth. And in that process, ethanol is produced, and more importantly, from the yeast perspective, NAD is produced. So you keep the fermentation process going. And in lactate fermentation, again, the type that athletes carry out, this is also the type of fermentation that is carried out in certain types of cheeses, when you're making like blue cheese and things. You have little blue specks and things that are in it. This is the fermentation process that's involved in those types of things. Also, this is used in if you know anything about so-called sour beers, they carry out a lactate fermentation as well. But at any rate, in this case, the pyruvate is at the end of this process. You produce a couple of NADHs. Once again, the energy from that is used to convert the pyruvate into lactate, and it builds up in your muscles if you're an athlete. Part of what causes them to ache after a race at times, or a volleyball match,
But more importantly, to your muscles and to the cheese, of organisms, it regenerates the NAD pluses so that they can be used. Fermentation produces 2 ATPs per glucose, and then it also produces either 2 lactates or 2 ethanols. There's a couple of other, again, in microbial biology, I talk about butane-diol fermentation and propionic acid fermentation. There are other products that are involved in the fermentation process with other organisms, but they are all for the exact same purpose. What is the purpose of fermentation? What does the cell get out of fermentation if it's anaerobic? NAD, exactly. It doesn't care about the alcohol. It doesn't care about the lactic acid. It doesn't care about the butane-diol or whatever other type of fermentation products come out of it. What it's after is that NAD so that it can carry out more glycolysis because it's using up the NAD. It's got to regenerate it. So about 36-ish are produced by aerobic respiration. As I've said, you'll end up with less than that in actual fact, but that's the official count if you would. I think I'm going to stop there. Yeah. So, Lorna, you're in charge of reminding me that metabolic pathways is where we start on Thursday, okay? Look at that. I owe you 7 minutes. How was your weekend? I went over to Lowe's and bought $250 worth of electric rotary saw, circular saw, and a battery and a charger, and a bunch of plywood. And I cut up enough plywood to make 7.5 honeybee swarm traps. So in the spring, I'm going to catch this and cook it. I'm planning on, right now I have 3 functional colonies. I'm hoping that by this time next year I'll have at least 10 or 12. So, I'm making enough honey right now for my family's personal needs and to give to family members at Christmas. You know, but yeah, it's sweet. Very sweet of me too. And they pollinate